Okay, this is chapter of microorganism and biotechnology. In today's lesson, we have to see the role of microorganisms in industrial biotechnology, how we can use this subject to make products at larger scale. The first product is that of penicillin, which is an antibiotic. For its production, we need to have a fermenter of a capacity of 100,000 to 150,000 liters. The product, which is penicillin antibiotic, is produced by batch operating system. What batch operating system is something I have discussed already in my previous lectures. The harvest or the product obtained, which is penicillin, of course, the antibiotic, is an excretory product of the fungus called Penicillium. So it's a very important point that you should remember that penicillin is the excretory product of this fungus. The process involves the addition of culture medium. This culture medium actually contains all the nutrients which are required for the growth of the fungus that we are adding into the fermenter. The culture medium includes lactose, which is milk sugar, organic nitrogen source, for example corn steep liquor, and we will add some mineral salts as well. The pH is maintained at 5 to 6, which is slightly acidic, of course, and the temperature is kept at 26 degrees Celsius. Aeration is done. Of course, the air is to be blown into the fermenter so that it is not only continuously stirred, but also oxygen is provided to the microorganism to respire on. Addition of strains of penicillium, then the medium conditions are maintained for rapid growth of this fungus. When the quantity of penicillin is maximum, the medium is collected. Penicillin would be purified by the process of crystallization. The next product that we have to know about is single cell protein. Before going into the process of how we will make it, we first need to know what it is. Single cell protein refers to total protein extracted from the pure culture of microorganism, for example yeast, algae, filamentous fungus or bacteria. So basically these are dried cells of microorganism which can be used as dietary protein supplement. They are used as animal feed and can be used for human feed as well as protein supplements. The process involves addition of culture medium into the fermenter. This culture medium would have glucose, inorganic nitrogen source, and mineral salts. The pH is maintained to slightly acidic medium. Temperature is kept at 30 degrees Celsius. Aeration again is done. Sterile air is blown into the fermenter. Keep in mind that no impeller or mechanical stirrer is used here as to protect the structure of the fungus. Then we will add the addition of Fusarium. It is a fungus which will be dried later and will be used for its protein content. The medium conditions are maintained. Nutrients are added when necessary for rapid growth of the fungus. The last product that we have made, which is SCP, is very important since it is used as a protein supplement. The question might have arisen in your mind that why don't we use protein in other forms like eggs, meat, or any other form of protein, why we have to use this one? So the answer to this is that SCP is purely protein. While if you consume protein from other sources, there are some other nutrients which would come with it. For instance, if you are using eggs as a protein source, the cholesterol level in eggs, in egg yolk, is quite high. So you are consuming egg if in whole, you are not only getting protein but also the cholesterol. Similarly, if you are using meat as your protein source, the meat also contains other things as well. It's not purely protein, it contains fats and fibers. Same goes for a protein which we are acquiring from plants. Fiber is coming with it, so we can't have a lot of pure protein from these sources. For that sake, we are using single cell protein, which is pure protein, gives you more protein per gram than any other sources of food.